Welcome to the PharmaSource podcast.
Today I'm joined by Berrade, Senior Director of Global Biologics Testing Sales at Charles River Laboratories. Charles River provides critical biologics testing services that help pharmaceutical and biotech companies navigate development and regulatory approval. UE leads its biologics testing solutions globally and will be exploring how testing strategy impacts development timelines, what's changing in the regulatory landscape, and how sponsors can build more effective testing partnerships to accelerate their programs. Thank you for joining me, Lewis.
Thank you.
So, you lead global biologics testing sales at Charles River. Just walk us through your background. How did you end up in the biologics testing space?
Well, first of all, I'd like to thank you for giving me this opportunity to join this podcast and explain how Charles River can support companies in the acceleration of the development of their drugs, and how they can bring their products faster and safer to the clinic or to the market. I'm a chemist by training. I have a PhD in medicinal chemistry. I completed that in Spain. After that, I spent a couple of years in the US doing a postdoc. And after that, I knew that I wanted to stay close to science, but I didn't feel like doing my own research. So I got the opportunity to join a CDMO in Spain called 3P Biopharmaceuticals. I was there for 3 years, always in business development related roles. And after that, I got the opportunity as well to join Charles River, the Biologics Testing Solutions group at this company. And since then, I've been in different roles, always in supporting like business development related and supporting clients to, again, provide clinical testing services that can help these companies to bring their molecules to the clinic or to the market. And the reason why Charles River was the right company for me is because it's a global company offering a very wide range of services globally, and that was really appealing to me.
As you say, obviously it's a huge global company, so you obviously operate across different regions and markets. How does the biologics testing landscape differ across geographies?
Well, in general, especially in the regulated markets like US and Europe and some other regulated markets, the testing requirements are pretty similar. Nonetheless, there are some, especially with the compendial methods, there are some differences according to the pharmacopoeias. So what we're doing, we're offering harmonized protocols that are compliant with both USP, EP, and in some cases as well, the Japanese pharmacopoeia. We're getting more inquiries to support as well, or to be compliant with the Chinese pharmacopoeia. So we're always trying to meet our client needs by offering these harmonized protocols that can be applied globally. When talking about semi-regulated markets, there are some differences, and in that case, we always are flexible with our clients just to meet their needs. But in that case, we need to understand from them what their regulatory requirements are and how they need to be supported.
And so biologics testing is obviously critical for development and regulatory approval. It's not always top of mind for companies until it maybe becomes a bottleneck. I'm not sure if you agree with that, but when should sponsors be thinking about their testing strategy? What's the ideal timing?
I know it's an easy answer, but it would be as soon as possible, as they know how their testing or the CNC package should look like. They should let us know. So ideally pre-IND, especially for most of the characterization and release methods, they should be thinking about it before they go into the clinic. Sometimes the challenges we face, or the challenges our clients face, are when they don't think about if methods can be validated and can be used for the release of the drug. So they maybe develop something in a research center, in a university, or they do the development in-house, and then they realize that they cannot validate that method according to ICH guidelines, and therefore they cannot use it for the release. So that's one of the challenges they usually face. The way we support them, we can, of course, optimize the methods. We can support them from a regulatory and scientific perspective to make sure that their method can eventually be used for the purpose that they have, which is usually the release of their critical materials, could be drug substance or the drug product.
What are the most common mistakes you see companies make with their biologics testing approach? What do they typically underestimate? Is complexity, timeline?
Right. I think in general, most companies have a pretty robust strategy, but also we also work with some companies who maybe didn't think properly about their phase 1 or phase 2 or clinical studies. So some of the mistakes are related to maybe vendor management. Some companies place different assays with different companies, and they end up dealing with several different providers for the release of the drug substance, and that becomes a nightmare. So that's something we can certainly help them because we can do pretty much everything under one roof. So that's sometimes one of the challenges we see. Also, in terms of the assay quality, sometimes we need to optimize their methods because maybe they didn't think well about the design of the study, or they didn't really conduct a proper quality by design approach. So those are some of the pitfalls or some of the, let's say, mistakes that some of our clients usually face during the early stages of their clinical development.
I see. And so in the last few decades, the biologics landscape has obviously evolved dramatically, from traditional monoclonal antibodies to ADCs, bispecifics, cell and gene therapies. How has that complexity affected testing requirements?
So, yeah, you're right. Over the last, let's say, 5 to 7 years, we've seen a clear increase in new modalities, including ADCs, gene therapies, cell therapies. So every modality might have different requirements. So in terms of ADCs, you need to have specific safety measures in your testing labs. For gene therapies in general, the principles are very similar to traditional biologics, but of course you need new methodologies, new assays. And then, of course, we see more urgency in getting the results. So timeline has become a critical aspect of the testing. For cell therapies, of course, timeline is critical. The shelf life of the product is very reduced. So what we see is that many companies need to do testing in-house, so we can support that as well, help them develop a method or validate a method that then they can transfer in. For maybe allogenic products, we can also support them with some of the rapid technologies like rapid sterility, rapid mycoplasma technologies available in the market, and we can do that. The goal is always to support the development and release of life-saving treatments and do that in a safer and faster way.
With that kind of complexity, regulatory expectations for biologics testing obviously continue to evolve. What are the key trends or changes you're seeing from agencies like the FDA, EMA, and others? What should companies be preparing for?
Well, I think the main change over the last year or 1.5 years has been the implementation of the revision 2 of the ACHQ5A guideline. With that new guideline, NGS has become much more popular, especially for viral safety, but also for genetic characterization, plasmid identification. So NGS, I would say, is the main breakthrough in terms of testing at the GMP level. Also, through this new guideline, companies can leverage prior knowledge for viral clearance, which is helping reduce the scope of the viral clearance studies as well as the cost and the material required. And then another important topic will be quality by design. According to the guidelines, there's an expectation that quality by design would be applied for method development, and that's going to become more and more critical. And again, this is expected by pretty much all agencies with a main focus on the FDA. In general, the agencies tend to be rather conservative in adopting new technologies, but in general, as long as clients can show that they have a robust method or assay and they can validate it, agencies will evaluate that in most cases positively. So hopefully that's encouraging for maybe companies planning to use new technologies.
With these changing regulatory requirements, with next generation sequencing, you said, and as an example, how do your labs keep up with those guidelines?
So we still offer the traditional methods. So we still offer most of the in vitro, in vivo biosafety methods, and we offer Sanger sequencing, which is, let's say, the traditional technology. In terms of NGS, our approach has been partnered with an expert in this field. It's a company called Pathoquest based in France, and they're really experts in NGS. So rather than investing from scratch, what we're doing is just partnering with an expert in the field so we can better support our clients. But we still offer the traditional technologies as well.
And you mentioned that regulatory bodies are a bit slow in adopting new technologies. What downstream impact do you think that has for, even patients ultimately?
Well, the impact for patients might be so by not using new technologies, you may run the risk of taking longer to get to the market or get to the clinic. So indirectly, that's going to have an impact into the clients because they might not have a potential treatment when they need it. So, but in terms of timelines, there may be a slight impact. Beyond that, I don't see a huge impact into the clients.
Okay. And so different therapeutic modalities often have different regulatory testing requirements.
Right.
How do you help sponsors navigate what's actually required versus maybe just what's nice to have?
Our experts have a really deep knowledge of the regulatory guidelines and of the science, and of course they are in close contact with key opinion leaders in the industry. So through that knowledge, we can help clients to tailor the program based on their molecule. Of course, the testing package for recombinant protein or an antibody or AAV is going to be different. So based on our experience, we can support clients to navigate through that process. Also, as a global company and a large company, we've been working with both small biotechs, retail companies, big pharma, CDMOs. So we've been exposed to many different situations. And again, in most cases, we can come up with the right approach to support clients depending on in what clinical stage they are and what kind of molecule they're working on. So we tailor our programs right along.
Yeah, as you say, you work with a huge range of companies from small biotechs to big pharma. How does your partnership approach differ based on company size and resources? For example, what does emerging biotechs need that's different from established pharma?
Right. In general, every client is different, but the trend we see with biotechs, they need flexibility. They usually have limited budget. They need to work with the CDMO, so they have we have to be flexible with our service. While what we see for big pharmas, they usually look for long-term partnerships. They look for a reliable partner that they can trust. They look for scalable capacity. They look for new technologies. And eventually pricing becomes as well a key decision factor for both the biopharma and the biotech companies. I think we've helped both. Small, as I mentioned before, small companies, big pharmas, CDMOs. So again, we try to support them in what they need, and we try to tailor our approach depending on their particular needs. But that's in general what we see in terms of the differences between small biotechs and big pharmas.
As such a big company, how do you ensure that small biotech companies are getting the kind of priority that they need, that they don't just become a number, but they actually feel like they're a priority?
That's true. I mean, we get that comment pretty often from small companies saying, well, you're a large provider, we're going to be the last in the queue. And actually, that's not right. So we want to be a solution provider, not just a service provider. So of course, depending on the relationship, we might need to work on the prioritization of some samples. But in general, we don't neglect small molecules. And actually, it's a big part of our client base. And they are as important as other big pharmas because their treatment could save a life, same as one product from a big pharma. So they're also important for us.
And so from your perspective, working with so many different clients, big and small, what makes a partnership successful?
I think it's trust, communication, an open communication. So the more information we get from our clients about what they need, when they need it, what information they have available, the better we can come up with the right scope of work and tailor our approach. So I would say in general, we need that trust. We need a really open communication and understand what they're going to need. And then we have a strong team of subject matter experts who are usually in contact with the CMC teams or the project coordinators at our clients. And they also can discuss about the technical needs of the program. So through that communication, that's how we can ensure that the program is successful.
Let's move on to talk a little bit about looking into the future. Your testing technologies and methodologies are advancing rapidly. What innovations do you think are making the biggest impact in biologics testing right now? Where is technology genuinely improving speed, accuracy, or cost versus just being different or, a bit of a hype?
I would say the main breakthrough in the last years has been next generation sequencing, NGS. There are different reasons why companies might want to use NGS for two different purposes, viral safety, but also genetic characterization. The main advantage, well, again, as I mentioned before, this is in line with the new ICH Q5A Revision 2 guideline, which is opening the door for the use of NGS as a replacement of some of the traditional methods. You can reduce the use of material, you can get way more information, and you can be faster and cheaper. So that's the main reason. There are other technologies like ddPCR or mass spec, which are also gaining momentum and will be used more and more for the new technologies.
There's a lot of talk about AI and automation and laboratory services. Where are you seeing those technologies actually creating real value in biologics testing? What's practical versus kind of just aspirational?
I think in terms of GMP biologics testing, it's more aspirational at this point than reality. I see a lot of potential in both AI and automation for drug development, drug discovery, drug development, preclinical studies, clinical studies. That's where I think we can really utilize AI and automation in a very powerful way, reducing the duration of the studies and the scope of studies and eventually the cost. In terms of GMP, I still see some limitations with a very few, well, some exceptions. Of course, for method development, especially method development, automation makes sense and it will help to reduce human errors. And there may be some advantages, but I think it's mainly upstream in the process development and product development or process development and drug development where both AI and automation can make a difference. Yeah.
As new modalities emerge and become more complex, how is Charles River planning to stay ahead of the curve on testing capabilities?
Our group of subject matter experts are really permeable and very open to evaluate new technologies, new pieces of equipment, methods that could be implemented to support not only new modalities, but also existing modalities that require better methods, either that require less material, that can release rapid results, and so on. So that's a key piece of that. We evaluate, along with our subject matter experts, our commercial team is in close contact with clients to evaluate what new technologies or pieces of equipment should be implemented in our labs. And actually, every year we acquire new equipment. So I think that's the keeping in contact with our clients, attending conferences, and talking to vendors is the way we have to stay state of the art in terms of the technology we offer to our clients.
Finally, then, let's just touch a bit on Charles River again. You know, with the biologics testing market being pretty competitive, various providers and specialized labs, how does Charles River differentiate? What's your value proposition?
Right. So, of course, we're a global provider with a very wide range of services. So we can support clients from their cell banking needs all the way through their GMP release testing, going through cell line characterization, bulk harvest testing, viral clearance, method development, qualification, validation. So we can support them every step of the way. So I think that's one of the key assets. Of course, having a global presence with labs in both Europe and the US is very powerful as well. I mentioned as well our scientific team, our experts, they can always jump in discussions with clients when there might be maybe some complex programs. And eventually it's just being a partner that they can trust. I think most of our clients have a really good experience in working with us. And as a matter of fact, many of our clients are very loyal to Charles River and they keep working with us. So we're grateful for that. And we'll, again, as mentioned before, we'll continue to stay ahead of the curve, implementing new technologies and making sure that we can help clients develop their drugs and bring them to the clinic faster and safer.
Fantastic. Well, thanks a lot. It was great talking to you.